Okay, so at this point, we have finished the overview of the digestive system. We have gone through all of the organs in the GI tract. Now we're going to spend some time focusing just on digestion and absorption of those three macromolecules. We've already talked about this a little bit here and there, but this just sort of emphasizes and focuses on that. So what are the three types of macronutrients? Those are carbohydrates, proteins, and lipids, and we're going to work our way through those, starting with the carbohydrates. As you work on this, I do recommend filling out the chart, and the chart can be found in the week 6 modules. So let's see, where is it? This one, macromolecule digestion worksheet. Okay, so go ahead and work on that. You can do it while I'm talking or at the end as a review. And I have already published the answers, so you can check it out first. Okay, so try to do it without the answers first. Okay, working on to carbohydrates. So when we are doing the digestion here, we're going to be looking at physical and chemical digestion. If we have chemical digestion, we're going to need a hydrolase, which is an enzyme that does hydrolysis, right? So this would be the process of breaking apart covalent bonds by adding in the water. Okay, so we're going to be taking our substrate and then turn it into a product with the help of a hydrolytic enzyme. Let's see, carbohydrates first. So carbohydrates digestion begins in the mouth. Okay, so remember in that saliva, we have salivary amylase. And what amylase does is it takes complex carbohydrates such as starches, it breaks apart the covalent bonds, and it digests them into smaller pieces. At this point, we mostly haven't gotten all the way to individual monosaccharides. We usually have disaccharides, but I want you to thinking about what this tastes like. So if you put a cracker on your tongue and let it sit in your saliva long enough, it's going to start tasting sweet. And that's because starches, they do not taste sweet, they taste starchy, but sugars do. So once you get down to simple sugars, monosaccharides and disaccharides, they are going to start tasting sweet. Okay, so it's going to start in the mouth, and then we're going to continue our digestion again once we get to the small intestine. Okay, so let's talk about why we skipped over the stomach. Okay, so you notice that we start in the mouth. That's by salivary amylase. Why does it stop once we get to the stomach? Okay, so please remember that these enzymes, most of these are proteins. This one is definitely a protein. And what happens to protein when you put them in pHs that are well outside their normal range? They denature. So this salivary amylase is going to denature once it gets down into the stomach, and then we have to reintroduce a new amylase from the pancreas once we get in the small intestine. So this pancreatic amylase does the same thing as the salivary amylase, it just has a different source. Okay? Okay, so here we have, so once you have your starch being digested by your salivary amylase, it'll digest it into disaccharides. From there, we need to continue our digestion all the way to the monomer. And what is the monomer for carbohydrates? That is going to be those monosaccharides. Okay, so the one I want you to remember again is glucose. Okay, so you take your carbohydrates, you break apart those covalent bonds, and that is going to be accomplished using those brush border enzymes. Remember, those are the enzymes that are embedded in the plasma membrane on those little microvilli. That's why they're called brush border enzymes. The brush border created by the microvilli has these enzymes embedded in it, and they continue the digestion all the way down to the monomers, which would be monosaccharides. Okay, into proteins. For proteins, we don't have digestion in the mouth, digestion of proteins is going to start in the stomach. Okay, so let's review some of those cells. We've got our mucosal cells that secrete mucus. We have the parietal cells that secrete hydrochloric acid. And we have the chief cells that secrete the enzyme that is pepsinogen. That pepsinogen is then converted into pepsin, and pepsin is the enzyme that is responsible for protein digestion. Okay, so all of this is coming in the gastric juice in the stomach. So digestion of proteins begins in the stomach. Okay, so let's talk about hydrochloric acid and how it helps with protein digestion. So the first thing is, because it is so acidic, when you put proteins in environments outside of their pH range, they begin to denature. And this is going to be very important because you're going to take that little folded, clumped up protein and unravel it. Okay, so this would be an example of physical digestion because you have not broken any covalent bonds yet. But it's important for the digestion because that does allow the enzyme then access to the peptide bonds, and then you're able to do chemical digestion. Okay, so the first way that the hydrochloric acid helps with protein digestion is denaturing the proteins, unraveling them so that they are more easily digested by the enzyme. The second is that the precursor, pepsinogen, is not active. It does not digest, but it can, in the presence of hydrochloric acid, be converted into pepsin, and pepsin is the active form of the enzyme, and it can digest the proteins. And also pepsin, it works best at its low pH. That's its optimal pH, and so it's going to work at the acidic environment that is created due to the hydrogen ions coming off of the hydrochloric acid. So very good, very, it's good to have an acidic stomach. We need that for our protein digestion. Okay, so in the stomach, you're going to take here's your unraveled, right? Your denatured protein, a long strand of amino acids. The pepsin is going to break them up into short little pieces, but it's not going to finish the job until you go into the small intestine. Okay, so from the stomach, we're going to go into the small intestine, where we are joined by two new sources of enzymes. The first is those brush border enzymes. Now, they're not the exact same enzymes that digest carbohydrates because the substrate is specific to the enzyme, but they're in the same location, so we call them the brush border enzymes. Those are going to be things like trypsins that will help finish digesting up those amino acid sequence, those polypeptides, into smaller ones, dipeptides, and even into their monomers, which are amino acids. Okay, so those are the brush border enzymes that are shown here. We also have proteases. Proteases are enzymes that digest proteins. Those are going to be found in the pancreatic juice coming in from the pancreas that is emptied into the duodenum. And that is going to, again, continue digesting those amino acids from their polypeptide forms into smaller pieces, which will be dipeptides, all the way into their monomers, which are those amino acids. Okay, so all of these little shapes that we see here, those are the amino acids, the small subunits for proteins. Okay, so at this point, we have talked about digestion of carbohydrates and of proteins. Let's talk about how we get those nutrients into the blood so that they can be delivered to the cells. So these molecules, they are polar. Okay, so think about how that influences cell transport. Okay, so think back to the cell transport. Because they are polar, they need a little help. So moving those molecules through the cells lining the small intestine, they're going to use carrier proteins. Okay, so they're going to use a combination of facilitated diffusion as well as some secondary active transport, right? There's co-transporters, and they're going to move through the cells. But the good news is because they are polar, they are going to easily dissolve in the plasma. What's plasma? Mostly water, and water is polar, and like dissolves like. So because the plasma is polar, these carbohydrates and proteins are also polar, so they easily just dissolve into the plasma so they can be transported to the cells in the plasma. And I say like that because it's going to become much more complicated when we talk about lipids, because lipids, and specifically triglycerides, they are nonpolar. Okay, so I'm going to go ahead and pause and start a new video.